That's so loud. Okay, so again, these are the muscle fibers or muscle cells. In this picture, you have 3, right? And you can see all the nuclei on the periphery, meaning on the outside of the cylinder, right? And you can definitely tell those striations. So if you took 201, you remember this. And we call it a striated muscle. Cardiac muscle is also striated. It's a little different, but that's they have the same type of arrangement. And then this one is the same thing, but it's a drawing, right? And these are the myofibrils, which have myofilaments, and those are the contractile proteins, right? That are gonna make this muscle fiber be able to shorten and lengthen, okay? So because it's a muscle cell and anything that begins with sarco means muscle, we call the membrane that surrounds the cell or the muscle fiber, we call it sarcolemma instead of membrane. It's sarcolemma. The cytoplasm inside is called the sarcoplasm instead of cytoplasm, right? Just because it's muscle. So it has its own little name, right? And you can see the big nuclei on each side. Just imagine that some muscle fibers or muscle cells, for example, that you have in your quads, they're long. Some of them are as long from your hip bone to your knee. So imagine, right, how long fibers are. So when we look at the ultrastructure of the skeletal muscle fiber and the typical arrangement, we realize that the fibers have what they're called sarcomeres. Sarcomeres, and it's right here at the bottom, the name, very little. Sarcomere is the basic functional unit of contraction. And you have to imagine the sarcomeres are one next to the other along the length of this muscle fiber and all around it because it's a cylinder. So basic functional unit of contraction is your sarcomere. So this sarcomere arrangement of these contractile proteins is what gives the appearance of striations right here on the top. So the figure on top is really an electron microscope, right? We don't really see this under the light microscope. This is as much as we can tell in a light microscope, which is what you probably saw in anatomy, or you will see when you take it. But this other picture is an electron microscope, so it's a lot more detail. And at the bottom here is the drawing of what we can tell from the electron microscope. So a sarcomere goes from a Z disc to another Z disc, okay? So here, this protein or this line that you see in little blue on the Z disc, this is an anchoring protein, okay? Anchoring protein. Then within a sarcomere, which goes from Z to Z, some people call it line, Z line or Z disc, it's the same thing. So this sarcomere is divided into the I bands, and you can tell that this is an I band made out only of what we call thin filaments, and you can tell that there's another I band on this other end. So from the Z toward the middle of the sarcomere, we only have, or at the extremes of the sarcomere, we only have thin filaments, and that's what we call the I band. Then in the center of the sarcomere, where we have thick filaments in green and thin filaments overlapping in the purple, that's what we call the A band. A band. Yes? Now in the middle of the A band, we have the H band right here. And if you notice, that one only has thick filaments. So what are the thick filament? Thick is called myosin. And the thin, which are the little purple ones, they're filaments of actin. Now, you probably remember that name when you learn about cells. Actin is one of the fibers of the cytoskeleton, okay? So the only way to remember the arrangement of the sarcomere is for you to draw it yourself. This is how you're going to remember it, right? So from a Z to a Z, we have a sarcomere. We know that we have I bands at each side of the Z disc going toward the middle. And those are only thin filaments. Then we have the A band in the center, which has both the thick and the thin filaments overlapping. So we have myosin and actin overlapping. And in the middle of that A band, we have what is called the H band. So why do we have to know this particular arrangement? Because contraction happens by what is called the sliding filament mechanism or sliding filament theory, okay? So this is on the overlapping zone, like you see here, this region here and this region here. You see that the myosin and the actin are overlapping. Just think that you have the fingers, right? So you have to have the right amount of overlap between the two so that we can do this sliding, okay? We're gonna see how that happens, but that's what happens. So if you think that my thumbs are the Z discs, right? And my fingers are overlapping here, this is the zone of overlapping. When they overlap, when they slide onto each other, the thumbs get closer. So that's what happens in the sarcomere. The Z disc get closer. So this shortens the sarcomere, right? Now, one thing that we have to have into consideration, imagine if you don't have any overlapping, your Z discs, my thumbs are far away. Can my fingers meet? No, I'm not gonna be able to contract here because there's no overlap. Think about what a muscle is overstretched, right? There's an injury, you did something and oh, you lost your overlapping. You can't contract, right? That happens. Or think about when you have a muscle cramp and they're like this. Can you slide anywhere if they're jammed like this? No way. So you have to have the right length of the sarcomere from Z to Z. It has to be just right because we have to have the right amount of overlapping so that we can do the sliding filament theory, okay? The shortening of the sarcomere indicates contraction. So if you think that, and I'll go back to this one. If you think that we're gonna have, let's see if I do it in maybe this color, brighter. So if you think that you have, that's one sarcomere, another sarcomere, another sarcomere. So my little lines are Z disc. So we have this, all this adjacent sarcomeres. And if you think about it, Julie cannot see, so I'm gonna do with black. Maybe you can see better. So think that we have thousands, hundreds of thousands of sarcomeres adjacent to each other, right? And they all are doing this, right? They're all shortening if we're stimulating them over the threshold, right? So the whole muscle fiber is gonna shorten, the whole muscle is gonna shorten. So that's what a contraction is. Is that clear? Yes, Julie. Julie M.
Okay, so if we go back to the other image with two, or the other slide with two images on the top, I see the pink, blue, and yellow, but I want to make sure The thin black line is Z discs, right? And then the thick
Yes.
black areas are overlapping.
Yes. So a sarcomere here will be, let me see.
Oh, is that a tiny little Z? Oh,
Yes.
I'm so sorry.
Yes.
I couldn't
So
see that.
it's from
Never mind.
here. You got it?
Yeah. I see them now. They're tiny little letters.
I know. And I have my glasses on and I can't see anything, but this is a sarcomere right here. Yeah. Okay.
Yeah.
All right. And you can tell that they're one adjacent to the other and all the way around in this cylindrical type of muscle fiber, right? It's amazing what we can do. But that's just the explanation of the sliding filament theory, right? This is what we think is happening. And I remember when we were face to face, I used to do this muscle contraction where you can actually hear the fibers slide onto each other. Celita, were you my student when we did that? Do you remember? It was so amazing. What did it sound like? What do you think?
I mean, it's been 7 years, but I do remember it.
Yeah, and so we used to put some electrodes on a student. We use the student as a subject, and then the student will contract his wrist and fingers, and we had headphones and we can hear, and it was like a whooshing sound. And some people would say, oh, it sounds like wind. Some people would say, look, it sounds like the ocean, but it was a whoosh sound. And that's the sliding of these fibers. Imagine that we can hear it with surface electrodes on the skin.
I'm not sure if anybody's heard it, but almost sounds like if anybody has had kids, like a ultrasound.
Yeah.
When they're moving and you can hear like the noise, like the heartbeat, when they're trying to check for the heartbeat, that's what it sounds like.
Yes. It's amazing. I love that. Just because once you hear it, you never forget it. So maybe.
Yeah, I remember now.
Yeah, you don't forget. It's like wind tunnel or liquid, kind of like whooshing. It was very, very interesting. I'm gonna try to find it because I think that I recorded it once. Okay, so here you have an enlarged picture of that sarcomere. So look at the myosin, which is the green thick filament, right? Myosin. So this myosin not only is thick and is big, but it also has all around it, think about a cylinder again, it has these little protrusions that are called the myosin heads. Myosin heads. Now these are important, okay? Because these myosin heads are going to attach to actin, which is the little double strand of pearls. If you notice actin in the purple, it's like a double strand of pearls. If you ever had a pearl necklace and it's double, and then it twists like this. So sorry guys, maybe you don't have pearl necklaces, but probably you've seen them. But they're globular proteins, globular round, meaning round. So it's just like a string of pearls, double stranded, and they're twisted. So that's what actin looks like. And because they're smaller in size, we call them the thin filaments. But you notice that those heads right here are going to touch the actin in several places. When they do that, when they attach, that's called the cross bridge. So I don't like that picture a lot because they're not really touching. Only when they touch is that we can call them cross bridge, all right? And that's important as well, because when these two attach and they bind, then with the help of, what do we need to contract our muscles? Energy. Yeah, Julie said it, I read your lips. energy. And what is our high energy molecule in our cells? What is it called?
Calcium? No, ATP. I don't know.
ATP,
Okay.
exactly. So when we have everything and we have that energy, the myosin head is going to do like a pivoting movement, or it's called cocking, right? And because it's attached to the top actin, it's going to move it with it, yeah? And that's what causes the sliding or those overlapping fibers to go toward the center. So much so that the H zone disappears when the sarcomere contracts. You can't see it, right? So a normal resting length of the sarcomere, you can see the H zone. But when we do the sliding, the contraction, the H zone almost disappears. So here you have the electron microscope picture, but the drawing is a little better. And that's how you can tell on number 3, how we don't see the H zone anymore, right? Because those fibers are sliding toward the middle and the H disappears. So this is how long a sarcomere is on number 1. A little bit of overlapping. We shorten the sarcomere, the H becomes a little smaller, and by the time you are on number 3, there's almost no more H, and the sarcomere has shortened a lot, right? The Z disc get closer too. So this is one of my favorite pictures. Or drawings, because it kind of like tells you everything you need to know on muscle contraction. So here we are focusing on one major myosin, which is all this green. And as I mentioned, the myosin has this myosin head attached with a myosin tail to the big fiber, central fiber here. But you can tell that these myosin heads are going all the way around, right? This myosin is a very thick cylindrical protein. Imagine all this is in the sarcoplasm of this muscle fiber or muscle cell, right? And we have tons of these ones. Then on top, you can see the actin, the double strands of pearls, right? And in this picture, we've added 2 more little proteins that they are associated with actin, but they are not involved in the contraction mechanism. So the 2 little proteins that are associated here is troponin, which are those oval yellow structures. So we have 4 in this picture, that's troponin. And then you have tropomyosin, which is the thin line in pink color. Y'all see that, right? And you can tell how that tropomyosin goes kind of like up and down, right? In this double strand of pearls. Now, troponin is attached to tropomyosin. So they're totally together, right? So if something happens with one, it's going to affect the other one. So tropomyosin is blocking the active site on actin. And that active site is kind of like an indentation where the head of the myosin needs to connect to form that cross bridge. So when we are in the relaxed state, muscle is not contracting, right? The tropomyosin is covering that active site on the actin, and the head of the myosin cannot attach. Now, if calcium comes into place, calcium ions come into the sarcoplasm, and that's where Julie had the calcium, right? Calcium attaches to troponin. And remember from Biosci 100, anything we add to a protein is going to change the shape of the protein, right? So by adding that calcium there to troponin, troponin is going to change shape. So think about troponin kind of like moving, and that movement, that changing of shape is going to move tropomyosin away of the active site. And that's how we can have this connection between the head of the myosin and the actin. So remember, once the head of the myosin attaches to actin, that's what we call a cross bridge. But then, in order to create that sliding mechanism, we have to have energy, right? We have to move. And anything that we move, we have to have that energy. So, oops, where did I there we go. Okay, so that's when ATP comes in, right? ATP, your high energy molecule. And I'm gonna tell you something about ATP that is gonna blow your mind. And I hope it does, because it did when I figured it out. Crazy. Okay, so look at ATP here, right? And look at the shape of this head of the myosin, right? But then when ATP hydrolyzes or breaks down into ADP and inorganic phosphate, that's when the head of the myosin does that movement, that cocking or pivoting, or the proper name is called power stroke. So think that of all the myosin heads that we have, when we can have a bunch of these heads forming these cross bridges, they're all doing that pivoting, that power stroke. Because it's a movement that carries that actin and everything goes toward the H zone, we need energy. Where do we get the energy? From ATP, right? Now you've all learned that ATP is a high energy molecule, right? Okay, now I'm gonna blow your mind. I'm gonna do my whiteboard. And so ATP, when it hydrolyzes or breaks down, this is the same thing, becomes ADP plus inorganic phosphate. But what else? It releases energy. So that's the energy that we use for cellular work. In this case, it will be for the power stroke, right? To do that pivoting, right? So ATP this way is what we call potential energy, right? Anybody remembers what potential energy is?
The potential energy is what's being held, like a battery.
Right, it's energy that we can use in the future, right? Potentially we can use it. Now this energy here is the one that we're actually using for work. So we call this one, anybody remembers?
Kinetic energy?
Kinetic energy. Yes. All right. So from what we've learned when we were not taking 200 level classes is that ATP is our energy molecule. Well, yeah, it is. It's a potential energy molecule. In order for us to use the energy, we have to break it down because we have to release that third phosphate. And only when we do that is that we release that energy that can be used for work, right? This is the one that we use for work. So are you all following me? Yes. So now let's go back to the picture that we had. And if you notice the movement of the myosin head, the power stroke, right? That grabs the actin because they're bonded together, right? It's happening when we are hydrolyzing ADP and phosphate. It doesn't happen when you have ATP on the other side. It only happens when you hydrolyze it or you break it apart because that's when you're releasing the energy, right? You're following me. All right, now I'm blowing up your mind. Let's go back to our whiteboard. I'm gonna clear all my drawings. So if you have ATP, head of myosin, releases from actin. This will be during relaxation. Oh boy, really bad my drawing. I'm much better in a real whiteboard, you guys. All right. ADP plus phosphate. This is when we have the cross bridge. Remember, this means the myosin and the actin are together. And we're doing the power stroke. Are you following me? Yeah. All right. Now comes the blowing of your mind. You need ATP to detach the head of the myosin and have relaxation. Isn't your mind blown? Didn't we say we need ATP to contract? Yeah, but not ATP. To contract, we need the breakdown. To relax, we need ATP in this form again. So you can go now and blow somebody else's mind, right? How do you remember this? So when we die, and I love those shows like Criminal Thing. I watch all of those. My husband thinks I'm crazy, but I like them. Because of the science too, because I can relate, right? I'm gonna hang it up on the wall, Chris says. Yes, please. So this is how you can remember, right? So ATP, detachment, relaxation. ADP plus PI, attachment, power stroke, contraction, right? So when we die, obviously no more oxygen is coming in. So we need oxygen to make ATP, right? So when we die and we don't have oxygen, let alone glucose for cellular respiration, the body stops making ATP. And if you don't make ATP, the head of the myosin stays attached to the actin, and that is contraction. And that is rigor mortis, right? Oh, I touched my rigor mortis. That's the stiffening of the body when we die, right? So if you watch the shows or police, they're also check for rigor mortis because you go and see the body is stiff. They can tell the time of death approximately, right? So rigor mortis will stay for about 12 hours. After that, obviously the body starts to soften, not because we're making ATP and we're detaching the myosin heads, but it's because there's decomposition and the proteins start to degrade, and that's what is the softening of the body, going all the way to decomposition, major decomposition. So is your mind blown now? I just want to know. Yay! So we need ATP to detach, to relax. We need ADP plus PI to attach, to contract, right? Oh, I love it. This is one of my favorite chapters just because of that. Any questions so far? Okay, my mission was accomplished. All right. So we'll go for a few more minutes. I give you a break because I know it's a lot. So here you have the hydrolysis of the ATP molecule. is releasing that third phosphate so that we get the energy to do the power stroke, right? And here you have the cycle of attachment and detachment. Of course, you can start anywhere because it's a cycle, but we start here on number 1, right? When you make the cross bridge, you have to hydrolyze the ATP molecule into ADP and phosphate. That can cause the cross bridge and the power stroke. That's what we have the energy for. And when we form ATP, we detach. We detach, and that means relaxation. Yay. Got it. Now, to all this, we now need to add calcium. We talked about calcium a little bit, but now we have to put it all together. So you can see a very close up of the actin, the tropomyosin, and the troponin. I don't care if you remember troponin complex, just remember troponin, right? And you can tell here how troponin is attached or bound to tropomyosin. So you can tell here that when tropomyosin is covering the active site, let me see, I don't know if you can tell there. Oh, my pen is off. Sorry, turn it off. Okay, so right here, can you see a little indentation on that actin? Kind of like convex. So that's where the head of the myosin needs to attach to it. But if you notice, the pink tropomyosin is covering that site. That's what we call the active site of actin. Say that 3 times really fast. So when calcium comes in, attaches to troponin, troponin, remember, anything we add to a protein moves or changes the conformation, and that is enough for tropomyosin to slide away of the active site, and that's how then they can form the cross bridge. The head of the myosin touching or binding to the actin. Yes, Julie?
Okay, so I had written that the tropomyosin blocks the active site on myosin, but it blocks the active site on actin.
Actin, on actin, yes, on actin.
But ultimately, myosin and actin are what we want to connect.
Exactly, yes.
Okay.
So when calcium comes in, and we're gonna see how calcium comes in, my beautiful picture is coming up. Calcium then attaches to troponin. It has a site for troponin. Troponin moves, kind of like changing the shape. And because it's attached to tropomyosin, tropomyosin kind of like slides away of the active site, and that leaves it free for the head of the myosin to attach to it. Remember that attachment is the cross bridge. The pivoting or the cocking of the head is called the power stroke. Now, how do we make calcium appear in the sarcoplasm? You've seen this before. This comes from nervous system. Skeletal muscle is what kind of muscle? Voluntary or involuntary?
Voluntary.
Voluntary, from our effectors. Remember when we learn about the reflex arc? So it's a voluntary effector. Therefore, it needs a motor neuron to touch, literally to be close, so that the muscle fiber can get that contraction. If we don't have that connection between a motor neuron and the skeletal muscle, the muscle cannot contract. So my picture here is the synaptic end right here, right? Again, the synaptic end, right? This little end here. Then you have the little vesicles right here, which are that hold the neurotransmitter, right? The neurotransmitter in the neuromuscular junction, NMJ, is acetylcholine. Now, acetylcholine in the neuromuscular junction is an excitatory neurotransmitter. So you can tell what kind of receptor we need to have in skeletal muscle. Nicotinic, right? Remember that acetylcholine can be inhibitory or excitatory. If it's excitatory, the skeletal muscle needs to have a nicotinic receptor. What's the name of the receptor where acetylcholine is inhibitory?
The G protein? Is that the
Yeah,
name?
it has a name.
Muscarinic or something like that.
Muscarinic. Oh, and we are going to go back to this one for cardiovascular, you guys. So don't forget it, right? This is when we start putting things together. So we call this the neuromuscular junction because it's my motor neuron on top that is attaching or very close to the skeletal muscle fiber. You see there's a skeletal muscle fiber. So I have a presynaptic motor neuron and a postsynaptic myofector or skeletal muscle fiber. So just to refresh your memory from the reflex arc, we are on this side of the reflex arc. Motor neuron, effector. So this is your chapter 12, you guys. Just that side of the reflex arc, right? So we had stimulus, receptor, sensory neuron, interpretation center, motor neuron, effector. So this side is just your neuromuscular junction. Good. Always referring back to our little reflex arc. So we go back to my beautiful picture. You have a lot of detail here, right? So we need a motor neuron. We need that motor neuron to be close to your skeletal muscle fiber. That motor neuron needs to be sending an action potential. So you can see the AP is the action potential, right? Remember what action potential with an excitatory event is happening. That action potential is causing a, what is this event right here where we are changing the resting membrane potential? That is called a wave of excitatory, chapter 7, a wave of
Depolarization.
Yay! Who said it? Who was that? Cassandra?
Me, Enriqueta.
Yeah, Ricky. Very good. All right. So a wave of depolarization is coming down. And remember, like we learned in nervous system, we have a little calcium coming in because we need calcium ion just so that we can release the neurotransmitter. Okay, this is extracellular calcium that it has to come in so that our neurotransmitter can be released in the synaptic cleft or that space, right? So we have acetylcholine then coming in, and you can see how acetylcholine attaches to this receptor, which we say is a nicotinic receptor, because we're trying to create another depolarization in the skeletal muscle fiber. When a presynaptic creates a depolarization on the postsynaptic, how do we call that potential? I help you with this.
EPSP.
Yay, excitatory postsynaptic potential. We want to create that on the skeletal muscle fiber because that's going to cause contraction, right? So we know now that depolarization is going to create contraction. It's an excitatory event, right? So if you look what is happening here, the acetylcholine is attaching to the nicotinic. And let me check the chat, somebody. All right, thank you, Anne. So the receptor is what? What is the receptor here? It's a sodium channel, right? So the minute acetylcholine attaches to this nicotinic receptor, it opens up and it allows sodium to rush in, and that causes a depolarization on the postsynaptic, which is going to create our contraction. Are we good so far? There's more. You can see these little tiny dots that I drew here on the postsynaptic or the sarcolemma of the skeletal muscle cell of fiber. And this is the enzyme that breaks down acetylcholine. So if you remember, it's called acetylcholine esterase. We can't have the neurotransmitter forever there, right? It will cause what it needs to cause on the skeletal muscle fiber, but then it will break it apart and then it's taken up by the presynaptic and it's gonna make more. So are we good so far with that? And this is the book's picture, which I think mine is a lot better. But what you can tell here is the, right at the bottom here, that's your neuromuscular junction in real life. And you can tell the fibers, the long fibers here like this. And then each of these little like feet is the motor neuron that we usually draw it like that, like one synaptic end, but in real life, it has several, I call them several little feet. So they're gonna be touching the skeletal muscle fiber in many different places. Okay, so that's what we consider the neuromuscular junction. One little foot and the area of the skeletal muscle fiber that is touching. Is that good? Any questions so far on this? No?
I do. I just want to clarify something.
Yes.
You originally had told Jolie, I think it was Jolie, that troponin and tropomyosin didn't really matter when it came to contraction, but it does.
Not really. They're just there bothering, really. They're blocking the active site on actin. So calcium helps us to remove it because the contraction is just myosin and actin. Now we have this little annoyance, which is troponin and tropomyosin, but those fibers are not involved in the contraction. The contraction, remember, is just the sliding, and the sliding happens because of cross bridge and power stroke. Yeah?
So it doesn't necessarily have to be released from actin, or that's what I think where I'm confused about, because when it's released from the actin, the tropomyosin is what blocks that active site on actin, right?
Yes. So they're not involved in the actual sliding. They're not involved in cross bridge or the power stroke, which is contraction ultimately. They're there blocking the binding site. So thanks to calcium coming in, we can remove it, and that's how myosin and actin can get together in the cross bridge and they can do the power stroke. So they're not involved in the actual contraction mechanism. They're just there bothering and covering an active site.
So, okay. So if we need ATP to release the myosin head for relaxation, what if tropomyosin doesn't block it? What if we don't have calcium to help block that?
Oh, if you
You know,
don't,
the active.
yes, in relaxation, you don't only need ATP so we can detach, but we also have to get rid of that calcium that is there. So calcium is going to be sequestered in the sarcoplasmic reticulum. That's where we store calcium in skeletal muscle fibers. And we're going to see that after. But remember the calcium that comes in at the synaptic end so we can release the neurotransmitter? That's what we call extracellular calcium. The one that we need for muscle contraction, it comes from intracellular calcium because we store it in that sarcoplasmic reticulum, which is what we've learned before as the endoplasmic reticulum. In the muscle fibers, they just act as reservoirs for calcium. So there's two things happening when we relax. We form ATP so we can have detachment. Remember, mind blowing. And we also need calcium to go back to the sarcoplasmic reticulum. And when calcium goes back, tropomyosin slits back and blocks the active site. Julie, M.
Not to exhaust the subject, but and calcium's role is to attach to tropomyosin, changing the shape of it and
Calcium attaches to troponin,
proponent.
which in turn is attached to tropomyosin. So kind of like they move together, right? Calcium, troponin, and since tropomyosin is attached to troponin, they're kind of like moving together.
Got it.
Yes?
Sorry, I think I helped confuse everybody.
No, it's okay. It's okay. It's just that the two major proteins that are involved in contraction is myosin and actin. That's it. Now, we have other little things that need to happen for that to happen, for those cross bridges to happen, right?
Okay, yeah, so they're just working together. I think that's what the confusion was for me.
Yes, yeah. They're not involved in the contraction, but they are there and we have to remove them so that we can attach those two major contractile proteins. So are we good so far? Okay, so I'm gonna stop for about 10 minutes because we've been going on forever. And then we're gonna put it all together. And then I'm gonna ask some of you to tell me the whole story. So be ready. The whole process from beginning to end. All right? Okay, let's go for a little break. 10 minutes. I'm back. Wait a little bit for you guys to return. Let's see. Yeah, most of you are here. So, what is important to understand is that we need a motor neuron to be literally, physically touching the skeletal muscle fiber. And that's the reason why if you have a spinal cord injury, this is your spinal cord, right? Depending on the height of the injury, some people could be not being able to move their legs, right? Hemiplegic. Or if the injury is high enough, that person could be a quadriplegic, right? They can't move the arms, they can't move the legs. And it's that connection of the motor neuron on the skeletal muscle fiber that is needed, right? If not, muscles cannot move. We cannot control or move our muscles. So the little picture down here, down below, is what is called a motor unit. So the contact of one little leg and the muscle fiber, we say that that was the neuromuscular junction, right? This is an N, okay? Now, a motor unit is a motor neuron. So we have my motor neuron here coming with the axon and all the fibers that it innervates. So if you see this motor neuron is innervating this fiber, this fiber, this fiber. So that's the definition of motor unit. Is the motor neuron and all the fibers that it innervates. That's the definition. So if you have very precise movements like our fingers, because we have to write, right? Or your facial muscles, because we have a lot of expressions, we're gonna have a lot more motor units. We're gonna have more than just one motor neuron because we need to have those fine movements, refined movements, right? But if you need to think about the quadriceps, for example, those are not refined movements. So we have less motor units for that kind of movement. So think specialized eye movement, for example, those are specialized movements that we have to have more motor units, which means more motor neurons innervating more fibers so that we can have those refined movements. Is that clear? I think that one of my discussion board questions is what happens to older people as they get older with the number of motor units, right? And as we get older, the motor units decrease. So that's why older people, unfortunately, we start becoming a little more clumsier. Things fall from your hands, right? In extremes, you will see some people that they shake a little. They're not very precise anymore because motor units decrease as we age. Okay, wait. So this is another of my favorite pictures, and this is going into learning how calcium comes out into the sarcoplasm and what causes the calcium to come out. Remember, we have to have calcium coming out because it has to attach to troponin, because it has to move tropomyosin so that finally we can have the myosin and the actin attach or form that cross bridge. So what causes calcium from to come out of the sarcoplasmic reticulum? So what causes it is the action potential coming down. So if you notice here, we have the same action potential that is coming down your motor neuron, right? And then we have the acetylcholine, the attachment here to the nicotinic receptor, right? So we're gonna cause a depolarization inside the skeletal muscle fiber, right? Now, these little squigglies here are sarcoplasmic reticulum. So this is what you all learn as endoplasmic reticulum. And I don't know if you remember, but the endoplasmic reticulum was a series of folded membranes, right? In just typical cells, we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Remember those two? So the rough endoplasmic reticulum is the one that has ribosomes and is mostly for protein synthesis. The smooth endoplasmic reticulum is more for adding glucose to sugars or for transforming some proteins. But in this case, in the muscle fiber, the sarcoplasmic reticulum is just to store calcium ions. So this is our intracellular calcium, right? So in muscle fibers, we store calcium in the sarcoplasmic reticulum. So this is different from that extracellular calcium that we need for the neurotransmitter to come out. Okay, different. It's the same calcium. It's just that this one in the synaptic end is extracellular. The one in our sarcoplasmic reticulum is intracellular, stays there, right? But it's sequestered in the sarcoplasmic reticulum. So when that action potential starts to come in and it causes this depolarization in the skeletal muscle fiber, the sarcolemma, if you notice, it's all made out of these invaginations. So this is how they actually look. Now, so far, we've been saying that your skeletal muscle fiber is a cylinder, but that's not the ultramicroscopic structure. In real life, the sarcolemma goes in and out on both sides. So that's what I have drawn here. So those are called the T-tubules. Another name for that is transverse tubules. Why do we have that? Because imagine if you had a cylinder, and I'm trying to look for a cylinder here. Let me see, what can I use? A big cylinder. I don't have a big cylinder. All right. I'm gonna get a candle, hold on. All right, so can you see the candle? Okay, so think that this is the cylinder, right? The cell will be long, but we're looking at it from this bottom part, right? So if I have an action potential coming in, if the sarcolemma was smooth like this candle, right? And the sarcoplasmic reticulum is that little black dot that you see there, which is the wick of the candle, right? There's no way that the action potential, that the depolarization right here on the surface can reach the center of the cell. And then in the center of the cell is where I have the sarcoplasmic reticulum. Got it? So we're going to be depolarizing the outside, but it will never reach that little dot, which is a sarcoplasmic reticulum. Now, if the sarcolemma is this way with these T-tubules in and out, when the action potential starts, it's going to go all the way deep and get and hit the sarcoplasmic reticulum. Get it? So in real ultramicroscopic structure, the sarcolemma is not smooth. The sarcolemma is like this picture. It's all with indentations, the whole cylinder, because we need to have that depolarization reach these little areas of the sarcoplasmic reticulum, and they are very deep. Remember, it's that little black thing there. Imagine. very deep. So that's the reason for having the T-tubules, because that depolarization reaches the sarcoplasmic reticulum, and that causes channels to open and dump the calcium in the sarcoplasm. So when the action potential reaches the sarcoplasm, the sarcoplasmic reticulum, calcium comes out. We're opening those channels, and for all those sarcoplasmic reticulums you see a bunch of calcium, and that's what we do with the calcium. The calcium comes out and it goes and attaches to troponin so that we can have that movement, so that we can get tropomyosin away from the active site. And that's how we can have the cross bridge form. So is that clear why we have those indentations that are called T-tubules? It's just so that the action potential can be deep enough to reach the center of the cylinder where the sarcoplasmic reticulum is. And only then is that we can cause that calcium to come out. So this is the book's picture of my picture. So I thought that mine is a lot better. Oh, I'm so confident. But you notice that this is the T-tubule coming down here. And of course it's called T because it has the shape of a T right there, right? But are those indentations that are going to create the calcium to come out? Because if not, there's no way the depolarization can get all the way in the middle of this cylindrical cell. Okay, I don't care too much about here. I just want to do this. Oh, what happened to my other picture that I wanted to show you? Huh. Did I miss it? No. Okay. I thought we had, okay, we have the, oh, this one is where we have calcium and ATP. So before we get to the other part of this chapter, I just want to recap all the different steps that we need to do or to have a muscle contraction, right? So the first thing that you need, number one, a motor neuron that is touching a skeletal muscle fiber. And that motor neuron needs to send an action potential that is excitatory, so it's a depolarization, right? The neurotransmitter is, oh, I forgot to record you guys. I'm gonna start again. Oh no, I never stopped. Okay, good. All right, so action potential that is coming as a depolarization from the motor neuron. The neurotransmitter in the neuromuscular junction is acetylcholine. Acetylcholine is going to attach to a nicotinic receptor on the sarcolemma muscle fiber. This nicotinic receptor is a sodium channel, so it's going to open and it's going to allow for a bunch of sodium ions to rush in. And that in turn causes a depolarization in the skeletal muscle fiber. Are we good so far? Now, the depolarization causes that deep action potential causes it to travel through the T-tubules so that we can cause calcium to come out of the sarcoplasmic reticulum and be available in the sarcoplasm. So now that we have calcium, calcium is going to attach to troponin. Troponin, because it's attached to tropomyosin, is going to change the shape and it's going to move tropomyosin away from the active site. And that leaves it free for the myosin head to attach to actin and that causes a cross bridge. Now, here's when ATP comes, right? The cross bridge has been created, but now we do the power stroke. So we need the energy there. And that's where ATP comes in. And where does it come in? Well, the sarcoplasm has a bunch of mitochondria, right? So we're constantly making ATP in our mitochondria. So that ATP needs to be hydrolyzed, broken down into ADP plus PI. And that's when we can do the power stroke. Now, whenever ACH, acetylcholine, disappears from the synaptic cleft, there's no more action potential. At that moment, we have to create ATP to detach the cross bridge. And at that moment, there are little pumps that are going to pump calcium back in the sarcoplasmic reticulum. And then we can start all over again. The cycle all over again. So who can tell me the whole story like that, like I just did? Starting with our motor neuron, right? And people can help. You can start and then get stuck, and you can, somebody else can continue. Is the more you repeat it, the better. The more it's going to get in your head as a process, right? And it's a cycle. And of course, the more you're exercising, or the more strenuously you're exercising, the more ATP you're going to need, right? So they say that each of our muscle fibers or cells have about 1,000 mitochondria. So imagine we have enough ATP, right? And it's sometimes if you're really, really strenuously exercising, mitochondria can split into two and make more right there to keep making ATP. Now, obviously, you know, for cellular respiration, what do we need? We need our oxygen, so we have to keep on breathing, right? Which is going to be a little more shallow breathing because you're increasing your respiratory rate, but you're increasing oxygen intake. But also you need glucose, right? So we store glucose in the liver as glycogen. So that's the reason why, if you think about athletes, before a big event, before a performance or something like that, they have to have a carbohydrate-rich meal, right? Because you want to make sure that this athlete is going to have enough glycogen stored that he doesn't have to stop and eat, right? So we can have that storage there so that we know we have a steady supply of oxygen, a steady supply of glucose. So this individual is going to keep making ATP so that we can keep breaking it down, so we can keep doing the power stroke and keep contracting.
That reminds me of when I was in high school and they would call them like carb loads the night before games.
Right.
And we now understand why.
Exactly, because you can't really, if you're running, if you're a soccer player, softball player, whatever you're doing, swimmer, you're not going to stop and eat. Oh, I feel like I'm depleting my glycogen. Let me stop and eat something. That doesn't make sense. The other thing I told you before is that the minute you put something in your mouth, your stomach thinks, oh, I have to digest this. So I'm going to divert blood flow to the stomach and the intestines, and I have to restrict it from other places in the body, right? We don't have enough blood for every single cell to get the same amount at all times. So we have to play, and we're going to learn that in cardiovascular. We're going to play with vasoconstriction and vasodilation, right? So if you're exercising and all of a sudden you eat something, then your body says, okay, I'm going to stop blood flow or less to the skeletal muscles because I now need to digest. So you have to divert some blood flow to those organs. And if you keep exercising, then you may get injured because your muscles now are not getting all the blood oxygen because now you have to take care of what you ate. So that's why you load up before the event. You load up before the big performance or run or swim or whatever. And then you eat after. You don't stop and eat in between so that you don't confuse your body, right? So if you have good sources of glycogen, you can go on for a while. As long as your oxygen is there and you have your glycogen, you're going to keep performing. You're going to keep running or doing whatever activity you're set to do, right? So I guess I didn't get any takers to tell me the whole story.
I can try if you guys want.
Look at my
I just
big,
wanted to give someone a chance.
okay, look at my big water bottle. Wait. You see that? Oh my God. Let's put it here. My Zoom background doesn't let me show you, but this one, oh, there, a little bit. So this one, it tells you at what time you're supposed to eat, drink. Look at that. You start at 8 and then you're done by 8 p.m. I love it. Okay. So Celita, you want to start? Nobody else is volunteering. Oh, baby's there.
He just woke up from his nap.
Oh,
Sorry, so I might be a little distracted, but I can try.
Well, you can start. Somebody can take over. So we start like there.
Okay. So, sorry. So then motor neuron, an action potential goes down a motor neuron, which then releases acetylcholine. Sorry. Sorry, guys. Which then releases acetylcholine. Yeah. And which then attaches to a nicotinic receptor and opens up a calcium channel and
Sodium
channel. And then calcium rushes in, which causes depolarization or an excitatory postsynaptic potential. I think it was. The action potential then travels down to the sarcolemma, which would be the muscle fiber. Okay, he's leaving. Hold on, sorry.
Yes.
Okay, so then it travels into the sarcolemma, which then travels down to the sarcoplasmic reticulum, only helped by the T tubules. This then causes the release of calcium into the sarcolemma. I think it is.
Sarcoplasm.
Sarcoplasm.
Yeah.
This calcium then binds to the troponin. I think that's how you say it.
Troponin.
Which then allows the tropomyosin to move and unblock the actin binding site. And then after that, the ATP is hydrolyzed, causing ADP and phosphate and then it releases energy, which is the kinetic energy that allows for the myosin head to create a power stroke. This causes a contraction, then ATP, then acetylcholine disappears from the synapse and goes back up to the presynaptic cell, I think it is, or neuron.
Yes.
And then ATP comes in and releases the myosin head, and then the muscle relaxes. Is that okay?
Yeah, and what happened to calcium?
Oh, calcium. I don't remember this part, sorry.
Calcium is pumped back into the sarcoplasmic reticulum.
Yes.
Hey, that was pretty good. That's pretty
Sorry for the baby though.
He came right in the middle of your speech. That's funny. All right. Well, Michaela says, clap, clap, clap. Very good, Salida. All right. So yeah, it has so many steps, right? In the only way that you're going to remember them is looking at the pictures, kind of like restate it every time, write it down, but then you know the whole process. If you try to memorize this, it's pointless. I would not even remember everything. But once you know the structure and how the actin and the myosin are, going through the steps and several times, if you need to write it down, if you need to record yourself saying it, that's the way that you're going to remember it. Okay. And it's good to start at the beginning, the motor neuron, right? Although I didn't start there, but I started with the sarcomere. All right. And then things that you probably have seen already or have heard, right? What is a muscle twitch? It's just one little contraction. That's a muscle twitch. And remember that I mentioned latent period. And if you turn your pen, that will be good. Latent period. So that's the time where the muscle fiber is getting ready. So then when it goes up, you have the contraction, and when it comes down, you have the relaxation. So the 3 periods of the muscle twitch, which would be one little fiber contracting, is latent period. Remember, here's when the fibers are creating tension, kind of like getting ready. Then you have the phase of contraction, and then the phase of relaxation. So that's how we handle a contraction or a muscle twitch. Now, what you see in the middle, which is called summation, this is when you stimulate right after the relaxation of the first contraction. So here you have the first one, and right after you stimulate again, maybe with a stronger stimulus, you can tell that the contraction is a little longer, and you can keep going, and the contraction keeps going up, and it's gonna go up to a point. So when you have that little picture of going up, making kind of like a little stairs, that's what is called summation. Summation. You're adding on to. So you're stimulating right after the relaxation. And sometimes you don't let it relax all the way. You can get to this, right? So depending on the frequency of the shock, frequency has to do with unit time, right? So if you stimulate 5 shocks per second, you're gonna get this. This is different from summation because you're not allowing full relaxation between the stimulus. So that's what we call incomplete tetanus. So summation, you stimulate after the relaxation. Oh, yeah. So contraction, relaxation, stronger stimulus, contraction, relaxation, stronger stimulus, and it's higher and higher. If you look at this one, you don't let the muscle to relax all the way before you apply the second shot and the third shot and the 5th shot, okay? So you start getting this little up, but it gets really difficult when you're doing more stimuli per second. In the second picture here, you have 10 shocks, and you can see how close those contraction relaxations are going on, not full relaxation again. And then if you give a lot of stimulation, you just get to complete tetanus. This is not good, right? Because if that happens and you continue the stimulation, you see how the muscle starts to decline, and that's when you get fatigue. Now, you get complete tetanus when you have an infection to the Clostridium botulinum. Has anybody taken micro here? Some of you? I think you have, some of you. So you get the same condition. The Clostridium botulinum, which is botulism, right? It releases a toxin that causes tetanus. So imagine people die because your respiratory muscles, they get to tetanus, complete tetanus, they cannot contract anymore, they're constantly contracted and they start to fatigue, and we need our respiratory muscles to breathe. So people die, they could die from botulism. So it's very serious, right? All right. So we have two types of contractions. The type of contraction depends on the actual movement that we're going to do. But isotonic contractions are the typical contraction that you could imagine. So walking downstairs, you had to bend your knee, right, when you're walking, or bicep curls. Those are typical isotonic contractions. And they can be concentric or eccentric depending on the load. So concentric is when the force is greater than the load. That's the typical type of contraction where you create force enough to lift a specific load. So this is kind of like the common type of contraction. The eccentric is that even if the muscle is contracting, but the load is so great that the muscle stops shortening and it starts to stretch. So imagine those bodybuilders, you know, in the Olympics when they have to lift those heavy, and they keep adding, and they're doing kind of like a lift, and they're like, oh, and they can't. That's an eccentric. They're trying to shorten the fibers. They're trying to lift the load. The force needs to be larger than the load, but they can. And at one point, the weight is so heavy that the muscle, instead of shortening, it lengthens. And then we have isometric contraction. Yes, Julie.
When you say that instead of shortening the muscle lengthens, is that an injury?
Well, it's not quite. I mean, they could, but not quite. It's just that they're trying to shorten, but the weight is so heavy that they start kind of like giving in. So that's what an eccentric isotonic. You're trying to lift, which is the typical isotonic, but the load is so heavy that they're not shortening enough to lift that heavy weight. So the muscles are shortening, but at one point they start to kind of like give in. So that's why they say lengthening, but it's not an injury all the way.
But they don't end up lifting whatever load. Exactly.
So that's where, you know, that the bodybuilders in the Olympics, sometimes I see they go through one round, then they go, oh, great, right? So they're doing a isotonic concentric because they're creating enough force and shortening to lift the load. And then they go to the next round and they add some plate, right? And now it's too heavy and they're trying. So they're starting the isotonic, and instead of becoming concentric, they can't lift the load. They can't create enough force. And that's when an eccentric is. They just can't lift it. And then we have isometric, which we don't realize that we do this one a lot, but it is there. We needed it as well when the muscles don't shorten, okay? So For isometric, the typical example is when you push your arms against the table, right? If you're standing and you push, you can feel the contraction. Even I'm doing it right now. If you push on your table, you can feel the contraction, but you're not lifting any weight. Or when you want to keep your head straight, right? I mean, the neck muscles, they need to be in isometric contraction. So isometric contraction is very important for postural muscles. So holding our head up, being able to sit straight up, right? Because we have a lot of isometric contraction in our erector spinae, the muscles that go along the vertebral column, that help us stay up, right? But you're not consciously contracting anything, or you're not lifting anything, right? It's just keeping your body erect. So that's what isometric is. Now we use both. The typical example is when you're going downstairs. In order for us to go downstairs, not only we're doing isotonic so that we can bend our knee, but you have to do an isometric in the back of your knees, in the hamstrings. So next time you go downstairs, do that. Or if you're a runner and you run downhill, you need to do isometric in your hamstrings so that you don't fall over. That's amazing, isn't it? So this one is, we talked about this one a little bit, that the length of the sarcomere has to be just right. We call this the Goldilocks principle, right? Not too short, not too long, just right. And the length of the sarcomere needs to be so that we can have enough zone of overlapping and the sarcomere can shorten. So if you look at the first picture to the left, this one, come on, yeah, I always do this. So if you notice in the middle, this is the right length of the sarcomere. You don't have to remember 225 micrometers, okay? So this is right or correct because it will give you the right amount of overlapping so that we can do the sliding, right? And in that specific length, you create the maximum tension, which is about that region of the graph. Now, when your sarcomere is too long, this will be when you overstretched a muscle, right? Look, we don't even have overlapping here. So you create no tension and you can't really contract. So that is serious, right? Because there's no way that you can contract. Or if you are on the other end and it's so jammed together that still you don't create tension for that one either. So it's either jammed or too far apart. It has to be just right so that you can do that sliding mechanism. So we talked a little bit about the requirements of the energy, right? So here we have to remember cellular respiration, yeah? And remember cellular respiration, where we first learned cellular respiration, it really starts in the cytoplasm. And that is when we only make 2 ATP net, right? And that one doesn't require oxygen. So we call that anaerobic cellular respiration, right? We only make a lot of ATPs if we go all the way into the mitochondrion and we use that electron transport chain. And that's when with the oxygen, we can create, I don't know, up to 38 molecules of ATP per glucose molecule. Remember that? So that process needs oxygen, or we call it aerobic cellular respiration, right? So when we talk about glycogen, right, and how we store it in the liver, right, and we can just break it apart, which is glycolysis. And then we need your GLUT transporters, which we mentioned on our first discussion board. And GLUT4 are the ones that are going to be taking glucose in the cells, right? So that we can use it with our oxygen and do our aerobic cellular respiration. So we want aerobic cellular respiration because we, having oxygen, we can make more ATP. That's pretty easy to understand, right? But sometimes if you only need to do a sprint, for example, you can just get by with anaerobic cellular respiration, right? Because if your exercise or your event is very short, like a swim dash very fast, or a sprint 100 yards, right? You can really burst with just the anaerobic cellular respiration. But if you think about a marathon that takes hours, you better have a steady supply of oxygen and a steady supply of glucose, because you're going to need a lot of ATP because it's a sustained type of exercise, right? So when you're exercising, you obviously start to increase your respiratory rate, right? So I mentioned before, oxygen or your breathing becomes a little shallower, right? It's faster, but you don't take that much oxygen with each breath. When you're resting, you can take more oxygen because you're not that fast. When you start exercising, your respiratory rate is faster, but you take less oxygen because your breathing is a little shallower. So there are other ways in which we can take oxygen in our muscles. Your legs, for example, have myoglobin. We're going to learn in chapter 13 that we have hemoglobin, that is the protein in our red blood cells that transport oxygen. But sometimes when we need additional oxygen, we can take it out of myoglobin, okay? Myoglobin is in dark meat. So if you think about chicken or turkey, a chicken leg or turkey leg is dark meat. And why is that? Because it has more myoglobin than, let's say, breast tissue, right? Why is the breast meat whiter? Because it doesn't have myoglobin. So dark meat, it has myoglobin, the white meat doesn't. So obviously, why would the chicken or the turkey have the red meat in the legs? Because that's what they use to move, right? So this is kind of like a little storage of oxygen to have just in case is needed. And for us, it's the same thing. Our legs have more myoglobin than other muscles in the body. Now, when you exercise, one of the byproducts of anaerobic cellular respiration is lactic acid or lactate. It's the same thing, lactic acid or lactate. So we need to break down lactic acid or metabolize it, okay? Because we have to get rid of lactic acid because lactic acid is an acid, and if it stays in the sarcoplasm, it may damage some of the contractile proteins, the myosin, the actin. So that's also the reason why, if you know you're exercising, we don't exercise the same muscles every day. You do upper body one day, you do abs the next day, you do lower body, and then you always have to leave a little bit of time so that we can metabolize that lactic acid, because having too much lactic acid accumulated from anaerobic cellular respiration can damage your fibers, and that's when we get injured, right? Another little thing that we have in our muscles, not only we have myoglobin in our dark fibers or dark meat, but we also have a little storage for phosphate. And why would that be good? Because we need phosphate to make ATP, right? Remember, ADP plus phosphate makes ATP. So we have what is called phosphocreatine in our muscles, and that's kind of like an additional source of phosphate. So I'm sure you've heard that some people take oral creatine because like that we can increase the storage of phosphocreatine. I'm not too, what would I say? I'm not too sure that the creatine that we take in supplements or powders, right? I'm not sure that it gets to the muscle. I mean, anything you put in your mouth has to go to your stomach, your intestines. You need to absorb it in your blood. From blood needs to travel to muscles. I have no idea what gets into your muscles after all that. But they said that it could increase your phosphocreatine storage. So we have additional sources of oxygen in the form of myoglobin and additional sources of phosphate to help us with ATP in the form of phosphocreatine. And that's what happens whenever we need ATP. We're going to take that phosphate from phosphocreatine and we're going to make more ATP. And that happens during exercise, and we can keep muscle contraction as long as we keep making that ATP, which remember, now you know, it has to be hydrolyzed in order for us to use it, right? So some of the reasons for muscle fatigue, you are accumulating too much extracellular potassium. So, you know, when some athletes, they will stop and eat a banana or half a banana or something. I've seen it a lot in tennis players, like in their little changeovers, they would just go and eat very fast, 2 or 3 bites of a banana. Banana has a lot of potassium, right? And when we exercise, because of the depolarizations, repolarizations, depolarizations, right? We lose some of that potassium that, if you remember, it has always to stay inside the cells, not outside. But if you lose it, then you're going to be having trouble with those depolarizations and repolarizations. So athletes will sometimes eat a banana really fast to kind of like replenish that potassium inside, intracellular potassium. You can also run out of glycogen, right? If you didn't have a good source or you didn't load up on your carbs before that event. Maybe you don't have a lot of calcium in your sarcoplasmic reticulum. These are all things that could happen when your muscles are fatigued. You accumulated a lot of lactic acid. That's a problem as well. There's a process by which we metabolize lactic acid, which is called, the Cori cycle. So when we start doing a lot of anaerobic cellular respiration and we're accumulating lactic acid, that lactic acid needs to recycle or be metabolized and it becomes glucose again. So, but the Cori cycle, which is to metabolize lactic acid and transform it again into glucose, it takes about 24 hours for us to do. So that's the reason that I explained before, that you don't want to exercise the same muscles every day, because you need to allow that 24-hour rest so that we can do the Cori cycle and we can get rid of that lactic acid. And the other thing, that if we accumulate lactic acid in your cytoplasm, it changes the pH. And that's a problem, because then there's a lot of enzymes and proteins that can be changing if we have a very acidic pH. Well, we can have lack of ATP, buildup of ADP, not having enough phosphate, and then it could also come from the motor neurons, right? So upper motor neurons, your central nervous system, we don't have ready and apt motor neurons. So that's why they say it's called central fatigue. It doesn't have to be with your actual muscle, but it has to do with the motor neuron that is coming down and trying to innervate the cells. This I already told you. So can we repair cells that are damaged? Well, it depends on the injury. Yeah, I'm sure that if you follow sports or athletes, when they have a very severe muscle injury, sometimes it's the end of their career. If there's some injury, we have some stem cells that are there from development, and they are close enough, they can repair some of the muscle fibers. But the injury doesn't have to be extensive. If the injury is very extensive, these little stem satellite cells cannot do the work. And what happens is that a lot of the fibrous connective tissue starts to invade the muscle, kind of like filling in where the injury occurred. And obviously the fibrous connective tissue is not muscle. So that muscle now cannot contract and relax the same way. So that's why there's some injuries that could mean the end of an athlete's career, right? If it's a severe tear of a muscle, then that connective tissue that we have from the epimysium, perimysium, endomysium, all that is connective tissue. So those fibers start to kind of like fill in the area of injury, and that could lead to not being able to perform the same way as before. And I mentioned this, what happens to conditioning or strength training, why the muscles would get bigger. And that is because there's what is called hypertrophy, which a little bit is good, it's nice, not too much, but a little bit is good. And this is the splitting of the fibers. So the thicker fibers can split into two, and that increases the size. So they kind of like open up, increasing the size. So a muscle that is used to be small can become bulkier or bigger. And that's what is called hypertrophy. And these are really neat. These are what we call the proprioceptors. And you should probably remember this from chapter 10. These are receptors that are located within the fibers and very close to joints, like here. So these are proprioceptors. In this case, is called the Golgi tendon. And here you have the muscle spindle. But these are proprioceptors, which means is that it gives your brain an idea of the position of the body, especially this ones because they're close to the bone. So when we move, that will give us an idea of our position. And the spindle, the muscle spindle, also gives you a measurement of the stretch. You know, when you're, even if you're doing a stretch after exercise, you know how much you can stretch, right? My Pilates instructor always says, stretch up to about 6 or 7, never to 10 when you feel pain. That's not a good stretch. When you feel pain, are these little receptors telling you, hey, you're overstretching, don't do it, you're feeling pain. So a good stretch is when you feel it, but it's not painful. And that's what these little receptors are detecting. And obviously receptors are there to protect us, right? Your eyes are receptors. They're protecting us from danger, right? You see something. You're overstretching. You have to know where up to stretch and not damage, for example, your muscle. So these are these proprioceptors that are there, again, to help and to keep us from danger. We've seen this one, your patellar reflex. When you pound on the ligament, that stimulates those receptors that are right in the middle, the muscle spindle, and the jerky reaction is your knee jerk. This one is the cross extensor. This is a nice one. This is when you have both sides of the spinal cord being activated, because the typical example is stepping on a tack. So when you step on the tack, what are you doing? You're lifting the heel, right? You go, oh, but what happens when you lift the toe, your foot? You at the same time, you need to contract your other leg muscle to hold you, because if not, you will be falling. So that's what is called the extensor reflex. So you lift one foot because it's painful on the sole, but at the same time, you need to hold your body. So your other leg needs to contract and be able to hold your whole weight. Is that, you understand that one? That's why it's called cross. Both sides of your, some people need to admit. So both sides of the spinal cord are being activated because you have to do both with your body, right? And let me check, did I have, yeah, question. Is botulism related to Botox? Yep, that's what they give you when you Botox injections, the toxin, but it's a very diluted, right? They're used to stop or how can I say this? Minimize wrinkles because they paralyze the muscles that are causing the wrinkles. Yeah, so they do a very diluted type of the toxin. Still is the same, is the botulism toxin, because what happens is that once you inject that, the muscle cannot contract anymore. And so you stop the movement that is causing the wrinkles. So some people say, no way I'm gonna use that. And then a little bit on the other two types of muscles. This chapter mostly covers skeletal muscle, but then we have cardiac muscle and smooth muscle. So the three types are skeletal, cardiac, and smooth, right? So remember, skeletal is the voluntary one, and cardiac and smooth is the involuntary, right? So on the right-hand side, you see the picture of a microscope for cardiac muscle. They're still striated, but they don't have peripheral nuclei. They only have one nucleus, it's in the center of the cell. And they also have these gap junctions that are called intercalated discs. So cardiac muscle is involuntary, is highly, highly branched. You can see here how the cells branched. And they have these little gap junctions that are very important for the depolarizations to travel to the whole heart. So think about skeletal muscle, right? Skeletal muscle is voluntary. And when we contract our arm, we don't have to contract our leg at the same time. So we can really isolate with skeletal. With cardiac, the heart is a unit. And when it contracts, it needs to contract as a unit, it needs to relax as a unit. So it's a little different. They still have sarcomeres, but they have these gap junctions that allows for the depolarizations to travel through the whole organ and have that contraction as a unit. So that's the difference. But they still need the calcium. They still need the ATP. They still have the sliding because they have sarcomeres. One thing, heart tissue relies more on extracellular calcium than skeletal muscle. So even though they have sarcoplasmic reticulum in cardiac muscle, they also have calcium coming from the extracellular space. So here's how you see calcium coming from the extracellular space. So you still have transverse tubules, right? You still have the sarcoplasmic reticulum here in green. Not only we get calcium from the sarcoplasmic reticulum, but we also get calcium from the extracellular space. So they rely on a little bit more of calcium. That's why they need that extracellular calcium coming in. Skeletal, all in the sarcoplasmic reticulum. Cardiac, sarcoplasmic reticulum, and extracellular. And then we have our smooth muscle. It's called smooth because it doesn't have striations. So it doesn't really have that very organized sarcomere structure. They have actin, they have myosin, but it's not arranged in a sarcomere. So it's called smooth because under the microscope, you can't see striations. The muscle fibers or muscle cells are kind of like spindle, they're called. So they're tapered at the end and a little more bulky in the center. So like here, you see the taper at the ends and a little bulkier in the middle. That's what they're the smooth muscle. But if you notice, the proteins, the thick and the thin, are in this little pattern that is mostly like this, the pattern of arrangement of these fibers. And you know what it reminds me of? You know when you buy oranges or lemons in those net-like baggies? You know that? So that's the typical arrangement of the smooth muscle fiber. And so the contraction is by pulling like this. So if you get one of those net baggies and you do this with a baggie, that's the movement that these fibers do because of that kind of like network type of arrangement. So they contract, they relax by moving this way. So skeletal and cardiac, we learned the sarcomere. Smooth muscle is this way. They still need energy, but it's very different because of the arrangement of these contractile fibers. And of course, where do we have smooth muscle? In every hollow organ in our body, right? So digestive, we're gonna see that we have smooth muscle. Coming up, blood vessels have smooth muscle. So with that, we can increase or decrease the diameter of the blood vessels. So we can do vasoconstriction or vasodilation. So the last slide is just a comparison between the three. And I think that I gave you enough information for you to know what are these the differences and the similarities between the three types. All right, any questions? I have to give a shout out to Celida for telling us the process. So good. So you have it. Too bad it's not for this exam. Okay, so yay, everybody's clapping. So we have a office hours on Thursday at 2. Then we have exam coming up on Tuesday. So it's gonna open Tuesday, 8 a.m. and it's gonna close on Wednesday at midnight, right? and it's 9, 10, and 11. So let me just check on. Oh, don't save all my scribbles here. Where's my canvas? I don't think I have it. I just wanted to make sure that we mention what are the chapters that we have to cover for the lab portion of the exam. So let me go to modules. Oh, I'm logged out, that's why. All right, so let's go and let me see if I can share this. Modules. Can you see my screen? All right, so let's go. We have chapter 9. We didn't have any labs for chapter 9 because we had our exam 1. So we didn't do a chapter 9 lab. So you're free there. Chapter 10, we did all our vision labs, right? So those are going to be included. And then for chapter 11, we did our endocrine, glucose, and thyroid. So you have 5, 7, right? That's it. So the vision ones and the 2 endocrine ones for exam number 2.